Good morning, everyone, and thank you so much for joining us for our seminar. My name is Emily Tom. I'm a fourth-year MD PhD candidate in Dorota Sclerosio Practice Lab, and it is my great pleasure to introduce today's distinguished speaker, Dr. Henri Leinonen, Academy of Finland Fellow and Adjunct Professor in Neuropharmacology at the University of Eastern Finland School of Pharmacy. Dr. Leinonen completed his bachelor's and master's degrees in pharmacology and then went on to earn his PhD from the University of Eastern Finland in the lab of Dr. Heikki Tammila. There, he studied electrophysiology of visual pathways as a screening tool for neurodegenerative diseases in mouse models. He then completed his postdoctoral fellowship at Case Western Reserve University and UC Irvine with Dr. Krzysztof Palczewski, studying the use of systems pharmacology to prevent retinal neurodegeneration, as well as homeostatic plasticity in the mammalian retina. Currently, Dr. Leinonen's lab is interested in the molecular mechanisms of homeostatic plasticity in the retina, as well as novel drug repurposing strategies for the treatment of retinal degenerations. In a recently published paper in Nature Communications led by Dr. Leinonen, they have shown that a treatment with a combination therapy comprised of 3 GPCR drugs slows progressive RD. And excitingly, these findings demonstrated mutation-agnostic efficacy and will facilitate further drug development pipelines. Dr. Leinonen's contributions to science have earned him several awards, notably several postdoctoral research grants from the Finnish Cultural Foundation, Fight for Sight, and the Eye and Tissue Bank Foundation in Finland. He was also the recipient of the 2020 Career Starter Grant from Knights Templar Eye Foundation. And in 2021, he was awarded a prestigious 4-year Academy of Finland Research Fellowship, which recognizes outstanding early career researchers. And it's truly our pleasure to welcome you back here today. Please give a warm welcome to Dr. Henri Leinonen, who will be presenting his talk entitled Systems Pharmacology and Drug Repurposing for Retinal Degenerations.
Thank you for the very, very kind, the best introduction in my career so far, Emily. And good morning, everyone. Yes, okay, so let's go to the pictures. So we had, of course, we said a lot of hard work, but also some fun times in the lab. And one morning in Case Western Reserve University, we had this photo shooting for Fight for Sight. And Chris was in a especially goofy morning that morning, goofy mood, and it was a bit hard to finally get a representative picture. But after some tens of shots or so, we finally succeeded to get something representative to be sent. I have no conflict of interest to declare, and here's the contents of the talk. So first, we will go through some definitions of concepts that I'm going to talk about so that you will be able to follow the presentation better. Then I'm going to explain why we target the intracellular signaling and especially why we use GPCRs as drug targets. And then I'm going to talk about our strategic approach to treat retinal degeneration and showing some proof of concept that was majority of it was published this summer. So systems pharmacology, I asked my friend ChatGPT and modified this answer a bit. So systems pharmacology is an interdisciplinary field that combines pharmacology, systems biology, computational modeling to better understand how drugs interact with biological systems as a whole. And unlike traditional pharmacology, which often focuses on a drug's target at a single target level, systems pharmacology instead takes a holistic approach, considering how drugs influence the entire networks of molecules, cells, organs, and even organisms. And some key aspects of systems pharmacology, one is that it looks at interactions within molecular networks to understand how drug affects complex pathways rather than just focusing on a single target. So this redundancy approach, focusing on single target, is kind of historical thing in the pharmacology field. So now we want to have a more holistic view. Systems pharmacology integrates data from multiple sources, including genomics, proteomics, metabolomics, data to get more comprehensive picture of drugs action, and it aids in understanding the complex mechanisms of disease by examining how drugs modulate network of interaction in disease cells compared to healthy cells. It is also used to identify new drug targets to optimize drug efficacy and to reduce toxicity. And some core components of systems pharmacology is the use of mathematical models and simulations to predict the effect of drugs. And then drug repurposing, also sometimes called as drug repositioning, basically means the same thing. This is a process of identifying new uses for existing drugs, and it leverages the fact that many drugs already approved for treating certain conditions might be effective in treating other diseases as well. I'm going to go through some drug repurposing strategies. First one, the use of bioinformatics and data mining techniques to identify potential targets for existing drugs. Me and my student Omar Semap is involved in a big project, and one paper is now under peer review. So this big consortium tried to evaluate all in silico tools that we have available for drug repurposing. So obviously these can be used to other things too, but this paper focuses on drug repurposing. So this consortium consisted of a lot of many experts from a multidisciplinary team, and it went through different in silico tools and created them so that it would be easier for biologists and other life scientists to choose from. So for example, for drug target interactions, there is in total 26 different databases. So we gave recommendations that what might be the best ones to use, and this hopefully will be published soon. So my student in the lab, Umar Semat, who is a bioinformatician, he's building up a method to simplify RNA sequencing analysis. So hopefully when he's ready, this can be done in a Windows in the Windows settings, so that the user don't need to know how to use command line coding, use command line, and don't need to know how to use Linux. And we plan to make this openly available, and this would facilitate drug repurposing. He's also building an ocular disease database that will collect genetic data, drug use data, and stuff like that from eye diseases. Another strategy is experimental screening, so testing existing drugs against a variety of disease models to find new therapeutic targets. So for example, we routinely test 2 different retinitis pigmentosa mouse models, not just one. And in IRDs, I think this is feasible because the disease progression is relatively fast in the mouse models and animal models, and there's a high translational potential. So if something works, it is quite highly possible that it can work in humans. Of course, this study design can be combined with in vivo injury models, so to say, like acute disease models, like bright light toxicity, sodium iodide toxicity, etc., and also can be combined to ex vivo kind of retina. And these experiments, I have a student working on this method, so organotypic cultures, so we can culture retinas, they stay alive for several days, and then we can expose them to specific stressors and study specific pathways and drugs. So the third one is clinical observation. So we don't use this, but this has to be mentioned because this is kind of the old way how drug repurposing was done. So now noting unintended beneficial effects observed during clinical use or clinical trials, and investigating them further. And the best known example of this is, of course, Viagra, that was originally developed as a cardiovascular drug, but you all know now where it's mostly used, for which indication now. So benefits of drug repurposing is of course reduced development time. It's cost effective, relatively cost effective. My favorite almost is that we know the safety profiles, so we know what kind of drug side effects this drug can cause. They will cause some drug side effects. All drugs in our world, in some patients, cause side effects. But the point is that we know what are the hazards and what kind of side effects they can cause. This is an important point. Also, regulatory advantages. FDA and EMA both have some programs facilitating the drug repurposing strategies. So really, we don't need to do so much preclinical testing with these drugs. So for example, this expensive toxicity testing don't need to be done at all, or don't need to be done so comprehensively, like teratogenicity, mutagenicity, and stuff like this. It's already done. Also, even clinical phase can be shorter. So a lot of these strategies would go directly to phase 2, don't need to even do phase 1. So this is the benefits, but of course, there is also some challenges in repurposing. Also, the dosage and formulation, the optimal dosage and formulation for the new use might differ from the original one significantly, and this requires additional studies. But of course, all of this can also provide rationale for patenting this new finding the new optimal dosage and formulation for new indications. So this video that I was showing in the side is a video of intravitreal injection in an anesthetized rat. And now, if we use an old drug, but we need to reformulate it, or we need to change the chemistry somehow, that the drug would stay longer in the eye, this would make it possible to patent this, either the medical device or the new compound, and that's how it could be also competitive in the market. So inherited retinal degenerations, then we can go this fast because global prevalence 1 in 2,000 roughly. Great majority of these are untreatable. Of course, we have Luxturna for RP65, but it's just 1 or 2% of the patients. This study, Australian study, it's an Australian study, but probably can be applied to industrialized world in the Western world, essentially. So this study concluded that, or found that IRDs actually cause most of the blinding certificates in working age people. So of course, AMD, glaucoma, diabetic retinopathy, they are more prevalent, but they only cause blindness in older adults and not so much yet in the working age people. And here's just the pie chart showing that most of this disease is IRDs, is retinitis pigmentosa. And there's a high heterogeneity. The inheritance patterns can be autosomal dominant, autosomal recessive, X-linked, and also idiopathic. I'm just showing this slide that there's a lot of different mutations, and there's almost most of these mutations we have an animal model, and in this presentation I'm going to talk a lot about RP10, P23H, and RP65 knockout mice. There's a lot of mutations, genetic mutations that can cause IRDs, as you know. So far, over 300 is found. So it's hard to come up with targeted therapies because of this reason. I mean, someday it will probably happen, but not in a very near future. And also the genetic cause is not known in 1 in 3 cases or even more, and not all cases are also monogenic. So this creates challenges for gene therapies. So why do we use intracellular signaling in as drug target in RD? It has been suggested that cyclic AMP levels may promote, elevated cyclic AMP levels may promote photoreceptor death, and they are a common feature in many animal models of IRDs. As a notation, also cyclic AMP suppression is being studied as a therapeutic target by others, and there's a good paper from 2018, and I know that this is being actively pursued. Also, intracellular cyclic AMP and calcium can positively regulate each other. This is quite complicated picture here, and it's a complicated system. And it's a very well known that calcium excitotoxicity is a common cell death mechanism. So this is a little advertisement. We got a paper accepted 5 days ago in molecular and cellular proteomics. My postdoc looked into proteomics of 3 different IRD mouse models, so the ones that we have used most, RD10, P23H, RP65. So nicely, there's 2 RP models and 1 LCA disease model, and we can look if there's something unique in RP, something similar between RD10 and P23H, and which is not necessarily found in the LCA2. When first we looked at what is common between all of these models, there's not a lot of protein pathways, not a lot of proteins, actually 23 commonly regulated proteins only, and pathways only 4, and phototransduction is, of course, one of them. And really, the proteomics in RP65 knockout mouse retinas deviate a lot from the other, from the RP models, RP10 and P23H. So the leftmost on these panels is the RP models, and then it's the RP65 knockout or LCA model, and there's a lot of difference in the regulated proteome. But then the last part of the paper we focused on, we did also like many different sets of proteomic experiments in RD10 and P23H mice, and we compared them to each other, and they are very similar. So we have a lot of differentially expressed genes that are same in these models, and how this is constructed, we always compare to wild type. So these heat maps tell what is different to healthy, and then we also looked that if these are similar, same proteins that are changing from the healthy state. So 600 proteins was common in these models, and 90 KEK pathways. So a lot. I cannot show all the pathways, but one of the most regulated one was the cyclic AMP pathway. So why do we want to use so-called old and boring G-protein coupled receptor acting drugs from all the possible targets that exist? So my main reason is really de-risking. So G-protein coupled receptors have a long history of use, and it's a very well characterized system. They do have a selective expression instead of widespread expression that it would be in all cells, like the same receptors, and this widespread expression doesn't allow selective targeting so easily. So, for example, ion channels, they are widely expressed, and kinases, etc. So GPCRs also, they have a modulatory action. They often act as modulators rather than inhibitors, or like full inhibitors or activators, and this modulation tends to create a more balanced therapeutic effect by gently influencing signaling rather than shutting down or overstimulating something completely. And this is kind of related to overstimulation. So there is built-in regulatory mechanisms such as receptor desensitization and internalization that helps to protect from overstimulation by agonists. Also, there's massive number of available GPCR drugs and compounds. I have drugs and compounds because we have a lot of approved drugs, but there's also a big library of drugs or compounds that pharma has developed, but they never reached the marketing phase. And it has been estimated that about one in 3 all the drugs that we use affect on GPCR. So it's a very kind of important family of receptors for drug targeting. So this paper appeared in summer 2016, just before my postdoc, and influenced a lot my choice of joining the Balczewski lab for postdoc. So in this paper, Chris and former postdoc of his and et al. They looked into gene expression profiles in the mouse and human retina and trying to figure out that which GPCRs are expressed in both of these species, so the species under study, and also the species that we try to invent the drugs for, so human, of course, and also to try to figure out that which of these receptors are easily targetable and already have a lot of good drugs. So dopamine receptors pop out, adrenergic beta 1, adrenergic alpha 1. So our approach was finally fully published in July this year. So I apologize it took so long, but there was a little bit of delay because I started my own lab here in Finland, and also some new stuff and analysis needed to be done also here. And then there was also other papers already before that kind of led to this whole thing. So we do use a multi-target approach. We are not targeting just one type of GPCR, but in this strategy that was published in Nature Communications, we do target 3 different GPCR receptors. So GS, stimulatory, GI, inhibitory, they converge into same system, adenylyl cyclase. So by inhibiting GS coupled receptor, we decrease adenylyl cyclase activity. By activating GI, we also suppress it. And this will effectively suppress cyclic AMP signaling, PKA activity, and also calcium influx into the cell. We also target a little bit different type of receptor, GQ, and this downstream signaling pathway culminates into decreased calcium release from ER. And we believe that all of these effects in the end culminate into decreased production of reactive oxygen species, among other actions. So, of course, we know the primary pharmacology of these drugs. They have been used for a long time. But of course, we didn't know before that do they necessarily decrease cyclic AMP signaling in the retina. And I collaborated with Shinya. Sato Shinya was in Japan at that time, and he had access to these mice that they called Pikachu. These Pikachu mice, they have it's a system to study PKA activity. So when PKA activates, there's more fluorescence. It emits fluorescence. And he put retinas under microscope and perfused these retinas, and he can put drugs into the perfusion. So first he was recording just baseline and then putting forskolin, which is a direct agonist of or activator of adenylyl cyclase, let it be for 1 hour, and then put the drugs and see if these drugs can prevent this increase in cyclic AMP and PKA activity. And this is a little video. Hopefully my PowerPoint don't crash now. So at 1 hour, he puts the drugs in, the forskolin in. Let's look at it again. So at 1 hour, he puts forskolin in. Photoreceptors turn completely red, so there's a lot of PKA activity. And at 2 hours, he also puts the triple treatment, and then we can see what happens in the photoreceptors. So goes red and comes back. Here's a still image of this and on the presentation. So we can see when forskolin goes in, we get PKA activity, and when the triple treatment is put back, also put in the incubation perfusion, it goes back to the baseline level, and here's a graphical description of that. Unfortunately, we were not able to do this with all of the monotherapies. It would be great to test them like individually, like bromocriptine, metoprolol, and damsulosine. He was able to do at least one experiment with bromocriptine. So now here the dose is the same as in the combination. It does suppress cyclic AMP activity and PKA activity, a little bit, but the kinetics is completely different and the effect is different. So one drug doesn't do the same effect. Now, because I don't have access to these Pikachu mice, we are continuing kind of the same concept though here at UEF. So my student Katri Vainionpää, a postdoc of mine, Ahmed Montasar, is working on this. So we use biochemistry and molecular biology methods to do the same thing. So we excise mouse retinas, do like an acute organotypic culture. We can activate adenylyl cyclase by forskolin and testing drugs if they can prevent the cyclic AMP increase. And indeed, in the first experiment, so this is just pilots and unpublished yet. So in RD10 mice, indeed, already at baseline, the cyclic AMP levels are up. And with forskolin, we can really increase cyclic AMP level by a lot. This is a logarithmic scale here. So now, interestingly, in this pilot, in a wild-type animal, promocryptin alone does decrease cyclic AMP signaling or turnover, but in RD10 mice not. So this is pretty interesting. So the system really in RD10 have changed because in a healthy state, one drug can do it, in the diseased mice not. So now going into actually telling the story of the Nature Communications paper. So TMB, so this tamsulosin metopro-promocryptin cocktail, slows cone photoreceptor degeneration in RD10 mice. And the system was such that the people who work with RD10 mice know that the disease progression in these mice is pretty fast. If you keep them just in a normal vivarium, by P30, all the rods have typically died. So what we did, we kept these mice in darkness. Darkness is known to slow down the disease progression by a lot in RD10 mice. And then treatment was started at P28. And we wanted to put the drugs in the diet. There's a lot of background and reason for that. We struggled for some time, like how to give these drugs chronically, but in the end, we used dietary administration. So the dietary administration cannot be started in P10 or something like this. It only can be started when the mice are weaned. So this was also one reason to keep the mice in the dark. So kept in the dark, treatment started, and then at P29, we put the mice back to vivarium. And then after 1 week in the vivarium already, the central retina is dead in the vehicle-treated mice, but less dead with the drug treatment. So more outer nuclear layer, there's some outer nuclear layer left, and also there's less retinal detachment. Here statistics to back that claim up, and Tim Lee was my student at the time. He's now an MD student in UCLA, was doing a lot of this ONL. We did tens and tens and tens of this, and also ERGs. But more than this, more impressive was the protection in the cone signaling. So these are photopic ERGs. This is a photopic ERG in response to green light stimulus. This is photopic ERG in response to UV light stimulus. And here you can probably appreciate that the cone signaling is over double, or at least double better with the drug treatment in comparison to vehicle. So at that moment, we also did bulk sequencing, so collected vehicle treated and drug treated retinas and run just a simple bulk sequencing. And here again, the comparison is first done to healthy wild type. So same age, same sex, wild type. And what does these heat maps tell? They tell the difference to healthy state. So the more bright the color here is, the more there is deviance. And we didn't focus in single genes or even single pathways or something like this, but looking at more comprehensively. So this is 5,000 more highest expressing genes in the retina and how much they deviate from healthy state. dark-reared P28 RD10 mice don't have that much deviance from healthy state, but then the vivarium, the mice that were put the vivarium at P36, the transcriptome is changed by a lot, but the drug treatment decreases this kind of like aberrant transcriptome. And we looked at this also in the mitochondrial level. We also looked at then also some more specific things like RP-associated genes. We also looked at catechol-aminergic signaling related, so adrenergic receptors, dopamine receptors, enzymes that synthesize catecholamines, and also this elimination. This I found really interesting. So COMT is an enzyme that helps tissues to get rid of dopamine and catecholamines in general. And in COMT expression in the retina is quite high, whereas monoamine oxidase is not so much. And this was really quite kind of interesting to me. So I also did test it with Western blotting. And indeed, I found that at protein level also in RD10 mice, COMT expression is increased. And I cannot think of any other reason why this is increased, that there is excess amount of catecholamines in the retina. But in the paper, we didn't focus on this thing too much. So it was kind of like a supplemental data. Okay, so we could show that retina signaling is improved, but indeed we want to test also for sure that their vision at the cortical level is improved. And I collaborated with Andrei Voigt's group, with him and his staff. So they did cortical recordings at 2 weeks on treatment, both local field potentials or VETs, visual evoked potentials, and the amplitude of these was increased with the treated mice. And also they recorded single cell responses, and in response to light stimulus, there was more spiking. Another good thing was that there was actually a little bit less background activity. So at light of spontaneous spiking was decreased in the drug-treated animals compared to the vehicle ones. So then at 1 month, and this was terminal experiment in these data sets, I did the retinal flat mounts, stained them against M and S opsin, and then we students in the lab, or my students at that time, was doing counting. So they were blinded to the treatments, of course, and we saw better preservation of cones. But again, the ERG is more impressive. So the effect in the ERG seems to be higher than in the anatomical markers. Some of the retinas I just smooshed and make homogenates and tested cone arrestin, which was higher in the drug-treated group, and also rhodopsin, and also optomotor reflex test indicated that visual acuity is improved with the treatment. So before the Nature Communications story, we also published other papers with light toxicity paradigm. And to me, this RD10, when we first keep them in the dark and we bring them to light, it was kind of like not as fast, acute light toxicity paradigm, but it mimicked a little bit the same. So then I wanted to test that, does the treatment have an effect also if we keep the RD10 mice all the time in the dark? So dark-reared RD10 paradigm. And this leads to very slow or quite slow disease progression. So first ERGs was done at 1 month on treatment, scotopic ERGs. So I didn't do photopic at that time because then they would get damage already from this photopic background light. So scotopic ERGs, and you can maybe already appreciate that this red signal here, these are group averaged waveform is increased, is a little better with the drug treatment compared to vehicle treatment, and here's statistics to back that claim up. An interesting point here is that anatomically, I cannot see yet anything at this moment. So we do have better signaling, ERG responses, but from OCT images, you absolutely cannot see any differences between the groups. This is actually, in a sense, a good thing, because this gives us an opportunity to study mechanistic effects. So what might the drug do so that when we look at longer, we see anatomical protection as well? Because if we do mechanistic study at the state when the retinas look different anatomically, I would say that we cannot conclude much about the mechanisms. So we focused the mechanistic investigation at this point. I was looking at lipid peroxidation assays, for example, 4-HNE ELISA, and 4-HNE was not increased in RD10 mice that was treated with the drugs compared to wild type, whereas this was double. 4-HNE content was doubled if the mice were not on drug treatment. So lipid peroxidation was decreased. This was kind of a secondary analysis of the same phenomenon, catalase expression with Western blotting and GFAP, where there's some trend of decline, but nothing really striking effect. So we did see a moderate cone protection also, but I have to say that I should have continued this experiment longer than just 3 months, because maybe you can appreciate that in RD10 mice, when they are kept in the darkness, there's actually not so much cone degeneration, even at age of 4 months. So the retinas look still pretty good, but in the central retina, there is some decline, and I did find improved preservation of M-cone count, but at this stage there was no difference in the cone ERG signals, but they were actually quite high still at this stage. Okay, so as I said, this gave us an opportunity because the retinas look similar. So at this disease state, even whether they are treated or not. So at this moment, we also collected retinas for single cell sequencing. And we wanted to see if we can find that in which cell classes there is most drug effects, at least in the transcriptome level, and really massive amount, hundreds of genes were differentially expressed with the drug treatment in Müller cells, but not so much in almost any other cluster. Another cluster that was a bit interesting was the cone bipolar cells and cones. In Müller glia, what was downregulated a lot, pathways that were downregulated a lot by the drug treatment was apoptosis, inflammatory response, nuclear factor kappa beta signaling, and JAK-STAT signaling pathways. So essentially just cell stress state was decreased, and upregulated ones included, for example, oxidative phosphorylation. Another interesting thing is, of course, this very different appearance of this string pathway. So downregulated, it's kind of just one clump, whereas in the upregulated ones, we have these like kind of specific clusters. So the beneficial metabolic alterations was found also in on-cone bipolar cells and cones with the drug treatment. So we found upregulated complex 1, complex 3, complex 4, complex 5 genes, and in cones also complex 4. And also what was also interesting in cones was this downregulation of some genes that are known to respond to elevated cyclic AMP. So this is probably just an indication of the primary pharmacological action. But also another interesting was that some genes that increase in response to increased dopamine, like CART-PT, was downregulated with drug treatment. Of course, when I had seen enough evidence that the triple treatment is effective, I wanted to check if we do get any beneficial effects with single drugs, and the answer is no. There was absolutely no therapeutic effect with single therapies. Then, of course, I needed to do all the doubles, all the possible doubles. And interestingly, tamsulosin and bromocriptine did not have any effect, whereas the doubles where there was metoprolol, so tamsulosin plus metoprolol and bromocriptine plus metoprolol, they were effective. So even the doubles can be effective if there is metoprolol, the beta blocker drug, although still the triple treatment was the best. So also retina function was improved in P23H rhodopsin mutant mice as well. At 3 months, kind of a similar story than dark red RD10. So in OCTs, we cannot see by naked eye any differences, but ERG signal is improved with the drug treatment. Here statistics to back that claim up. When we do statistics, when we do a lot of the ONL analysis and ANOVA statistics across time, there is some statistically significant effect, but it's very small. So again, the function is improved better than the anatomical hallmarks. And here's a photopic ERGs that were originally the same between the groups, but with time, the drug treatment group had higher amplitudes, both in M cones and UV cones. So also, of course, doing the flat mounts at the end of study, this was a longer study. The study was ended at the age of 8 months, and flat mounts was done, again, all the counting and so on. And there was a modest effect on S-cone population, but not M cones. And the retina at this point was really completely or quite gone. So we cannot see outer nuclear layer really in the OCT. So essentially just cones left. So this was the most surprising thing to me during these experiments and during these years, that this treatment was also effective in the LCA model, RP65 knockout mice. So this was super surprising. So almost as a joke, I put them on the diet, but I was quite surprised to see that the treatment was effective. So ERG signals, it improves sensitivity, scotopic ERG sensitivity, because in vivo, in RP65 knockout mice, we cannot see a wave at all. We were extremely curious to see if the treatment affects the photoresponse, so photoreceptor response, or then the inner retina signaling. So we collaborated with the Kefalov group, and Alexander Sasha Kolesnikov did these ex vivo ERGs. So as you can see, these are the photoresponses. These are just representative ones. But when synaptic blockers are used, we can isolate responses so that it's only coming only from rods, and we don't see a treatment effect at all. But then when the blockers are not used and we see the whole ERG signal, we get a B wave, and the B wave is increased with the drug treatment. And by the way, this was not an acute, just an acute treatment with the drugs, but these mice had been treated for 1 month, and then in the perfusion, there was no drugs anymore. Also, the treatment improves optomotor behavior. We have better contrast sensitivity with the drug-treated animals. Not so surprisingly to me, at least, the treatment was not able to slow down rod or cone degeneration in the RPE65 knockout mice. So ONL thickness and M cone counting, there's not UV cones anymore essentially left, but M cones there is, but the count is similar between the same between treatment groups. So doing again the same studies essentially as earlier I showed with RD10, so doing bulk sequencing, RNA sequencing, and comparing to wild type first, and doing heat maps and seeing the deviation from healthy state, and there's less deviation with the drug treatment in nuclear genes, also in the mitochondrial genes. And catecholaminatic signaling now is quite interesting. So these are regulated also in this model, but actually less with the drug, with the TMP drug. And mitochondrial function was quite striking. So it's changed a lot in RP65 knockout mice when treated with vehicle, but the difference from healthy state is much smaller with the drug-treated animals. So also we then decided to do single cell sequencing also to see, is it again Müller-Clear where the drug effect is coming from, at least in the transcriptome level, and indeed it is. Müller-Clear had the highest number of differentially expressed genes between the vehicle-treated and drug-treated RPE65 mice. Another interesting things here is the rods and rod bipolar cells that we look next a little bit in detail. So in RPE65 knockout mouse rods and rod bipolar cells, we see upregulated a lot of upregulated mitochondrial genes, these empty genes in both sides. And these ERGs, these are in vivo ERGs now, actually are from the same mice than this single cell sequencing was done. And we really get improved sensitivity. So the maximal response is not anymore so much changed, but so much different, but the sensitivity is improved. So again, we did also in this mouse model, the monotherapy and the doublets monotherapies were not effective, similarly as in RP10. But with the dual therapies, metoprolol and bromocriptine treatment was effective in this model also. Finally, we also did a 7-month-long study in a large animal model of RP retinitis pigmentosa PDE6A. So these animals have a little bit similar phenotype than RD10, although this is a little bit more robust. So almost mimics more like RD1 mice, sorry. Of course, we did pharmacokinetics here. And by the way, the treatment was not in dietary administration now. It was not feasible in this context. So we used altered infusion pumps that were changed every 2 weeks. And then again, Jinue was doing LC-MS from serum samples, and we checked how much drugs we have in the plasma, in the serum. And as just a summary, we did not get good enough dose for metoprolol. So we got good, okay dose for tamsulosin and bromocriptine, but metoprolol should be in the level of 100 and up nanograms per milliliter. But we tried to increase it in the dog studies, but it was hard. But still, we did see some positive effects. So ERGs was recorded. Photopic ERGs was recorded every month in the study, and we did see a kinetic shift in the B wave. So B waves occurred a little bit earlier with the drug-treated animals. It was not a massive difference, but it was statistically significant. Unfortunately, we didn't see effect in the amplitudes. At the end of the study, again, flat mounts was done, cone counting was done. This is PNA staining, and we did see an improved cone population in the superior middle retina, but not in the area centralis, for example. And all of this in vivo PDE6A studies was done in Simon Peterson Jones lab in Michigan State University. So zooming into some of the ERGs, the first recording you can see here, and maybe you can appreciate that it's a little bit left shifted, the drug-treated response or the group average response. So it's a little bit faster. Nothing striking, but there is some effect, because this is not very variable in ERG, so it occurs at the same time. Also, maybe you can appreciate that in the last recording, we don't get statistically significant effects, but just qualitatively, we can see that the red response here that represents the drug-treated group seems to be bigger. So now, 10 years later, not 10 years from starting my postdoc, but 10 years from this science signaling project, essentially, we get now these single cell sequences for free. So people do put this in the databases, and it's easy to use. So we can look where these cells are really expressed from big data sets. So not just 1,000 cells or 2,000 cells, but like tens of thousands of cells. And let's look into ATRA1A, which is tamsulosin's target first. These are UMAPs. And these receptors are expressed in Müller glia, and they are expressed in bipolar, some of the bipolar cells in mice. Then adrenergic beta 1, which is a metoprolol's primary target, it's expressed in bipolar cells, in some specific amacrine cells, and interestingly in cones. And then dopamine receptor 2, which is a promocryptine's prime target, it's expressed in the same bipolar cells as, so there's like a co-expression-like phenomenon with the adrenergic beta 2 and also same amacrine cells. So as a summary from this Nature Communication, focusing mostly on this one paper, so according to gene and protein expression, the catechol-aminergic system may be overactive on RD retinas. And this combination therapy's therapeutic effect on retinal function exceeds the anatomic photoreceptor protection. And we conclude that the effect of the drug treatment is unlikely directly via photoreceptor action, but it may include more complex interaction with Müller glia and inner retinas to resuscitate the entire tissues. This is a picture from this February in Costa Mesa. It's always nice to come back and meet you guys. So hopefully I can do that soon again. So thank you all. Thank you, Chris. Thanks the lab. So I named some of the people who I feel like affected my career and my postdoctoral research a lot at that time. I had a lot of good discussions with Philip Kaiser, who was my pharmacist colleague in the lab. Of course, big thanks to Simon Peterson-Jones and the laboratory for all the animal studies. This is a picture from postdoc times in UCI, I think year 2018. This is my current lab, current funding and a previous funding. And with that, I would like to thank you for your attention.
Thank you, Henri, for your talk. Chris, do you have any comments, questions?
No, I don't have any comments except of congratulations of the mass amount of work that Henri did, and with the quality of the work. Always the quality adds a value because it teaches us seriously about the problem. So Henri, congratulations. That was a wonderful presentation. Superb.
Thank you, Chris.
So I'll start off the questions. Do you think that for people with IRD, that, for example, if they have to take BMT indefinitely, or do you think that a early intervention for a little bit of time is enough to stabilize the disease?
Yeah, no, I think it would be for life. So indefinitely you will need to use it. It will not, like, this is the same with any chronic disease. So if you have a heart failure or elevated, let's say glaucoma or something like this, it's a chronic disease. With this type of therapies, you definitely have to use it the rest of your life.
Hey, Henry, this is Vladimir. Beautiful work and good to see you doing so well. A couple of questions. I'm very excited about your results showing that cone function or cones might also be preserved with your triple treatment. So have you explored further looking at actually the morphology? You showed some ERG data, but have you explored to see whether cone structure is preserved? And I would also encourage you to look more and find a cone rod dystrophy model so that, again, you're not saving cones because the rods are not dying. And look more directly at the preservation of cones, because I think they're, in terms of human vision, that would be amazingly important if you can preserve cone structure.
Yeah, thanks for this very good question. We did not in detail go and look into cone morphology, so we just did like a cone counting, the number of cones. But this is actually a good idea to look into go look into detail how the morphology looks like. Although I have to say that after all of these things and after analyzing all the massive amount of data that we had, and I also indicated this in the talk a bit, it seems that we do improve inner retina signaling much more so than the photoreceptors. But still, yeah, I think this is a good next direction to look into.
Okay, that actually is a perfect segue for my second question. I don't remember if you've looked at ganglion cell survival or other cell types, so maybe RP cells in other disease models with the same treatment.
No, we haven't done that. So there was this earlier project that I was partially involved in, published in Molecular Pharmacology, the same treatment in the diabetic retinopathy. And it did have a therapeutic effects also on the vascular leakage and also oxidative stress and these biomarkers. And it's very interesting. Hopefully somebody does it who focuses on glaucoma. But
Okay.
Yeah, so far we still putting all the effort in IRDs. I actually try to really push this to the clinical phase 2 trial.
Thank you. Great.
Hello, I'm Dr. Leonin. My name is Jacob, and I'm in the Brown Lab. And my question is, during your experiment, did you notice any systematic side effects to the medications you were giving to as your triple therapy? As all three of these medications at therapeutic doses can cause changes in blood pressure, and wondering whether any of your animal subjects started experiencing health effects from that.
Yeah, so thanks for this question. This was, of course, super important point that if we do cause some systemic effects, if we would cause some systemic effects on the treated group, it could be that they are just more stressed, and this stress could affect the disease course. So this is extremely important. So the best way to evaluate overall health in rodents is to look at body weight increase. So how do they eat enough? And we did that, and there was no differences between the groups. And I didn't see any obvious side effects. One peculiar effect that I saw in RP65 mice, and I don't know why this is in RP65 mice and not in RD10 and P23H, but they were grinding the pellets. So because we had red color in this, this was color coded, the drug pellets, and they were grinding the pellets more than the other animals. And I don't know why. It was really, really, really strange. But this was the only thing that we could distinguish these groups, that what they do different. And this was only specific for this one mouse line. And all of these mice are C57 black background, but still only RP65 knockout mice were doing this. So I don't know if because they are congenitally completely blind from the birth or almost blind. So maybe their taste is so good. So this kind of sensory substitution, and they taste the drugs more than the other models. This is just speculation. I don't know, but this was the only kind of weird effect that I saw. Yes, I think online there's questions.
Hello, this is Eric Hartman with the Kareuter Research Foundation. And given the prevalence of CHM in Finland, you had talked about the building of your computer models using the databases that you got there of IRDs. Do you see any potential benefit here for CHM research?
Hopefully, I don't know. So the challenge in Finland is that the population is quite small, and when we talk about rare diseases, and this is also poses a challenge to my urges to do a clinical trial here, because just have like a handful of patients. But yes, I mean potentially, hopefully. So we will see. I'm not a clinician, so I don't know how many patients we have. So
I know that the prevalence in Finland is about 1 in 8,000 versus 1 in 50,000 on a global.
Okay. Yeah, 1 in 8,000. So there's some hundreds of patients. Yeah.
Okay.
Yeah.
All right. Well, thank you.
Interesting point. Yeah. Thank you.
Hi, Henri, it's Emily. Good to see you, and congratulations on all this wonderful work. Can you talk a little bit about your rationale for going for the systemic administration dietary route versus directly going to the eye?
Yeah, great question. Of course, we would like to put it in the eye. Like ocular treatments would be best administered locally, eye drops or something like this. But simply small molecule compounds, even if you inject them inside the eye, they will stay there for an hour or something like this. So you would be needing to inject drugs into eye all the time. From eye drops, then most of the drug will never reach the retina. Like you can treat anterior chamber, so you can treat glaucoma with drops, but I mean, by estimation, if you put one drug molecule in the cornea, if you put 1 million compounds in the cornea, only one of the molecules will go reach the retina. So in humans, it's just almost quite infeasible to treat the retina. Dietary treatment, we chose dietary treatment. We tried different types, systemic treatments. We tried infusion pumps, but this is quite stressful for the mice. They might need to be single housed, etc. We also put drugs into drinking water, but the drugs were not chemically stable in water long enough. Then we were also doing the conventional injections, but the half-life of elimination of these drugs are very short in rodents. So 2 injections a day is not enough to keep a stable level. So in the end, we ended up to pellets, and hopefully, fortunately, it did work.
So going back to the RP65 knockout animals, can you speculate as to why the sensitivity increased even when you didn't preserve anatomical function or rescue saw death?
Yeah, so my best guess is that we resuscitate the inner retina. So it's actually like as you saw from these ex vivo ERG signals, we did not improve photoreceptor function, but we did improve the bipolar cell response. So I think we are resuscitating the surviving retina. And also when we looked at the single cell sequencing, this could relate to improved metabolic state. This is my best bet. Of course, I cannot know for sure, but this is what the data indicates.
All right. If there are no more questions, last call. Let's thank our speaker one more time. Thank you, Henry.
Thank you, guys. Have a nice weekend.